All right, everybody, welcome back. So this particular lecture is going to be mainly on epidermal variables, right? We're going to go back to this area of epidermal variables. As I said, not particularly my area of research, but I'll do my best to explain what the fundamentals behind the epidermal variables. We're mainly going to be looking at sweat-based epidermal variables, the platforms, the materials that are used, the instrumentations that you require, and the applications in this particular series we have here. Okay, so in terms of course structure, this was actually meant to be for tomorrow. That's where you saw the different date on the main screen. We've just brought it forward because I want to keep that dual receptor thing, which involves combining bioreceptors lecture for tomorrow morning. So I've just swapped the order around a little bit, but it's still going to be considered as lecture 5. Okay. So the learning objectives for this particular lecture are mainly to understand what the composition of sweat looks like, what's its relevance. We've already touched on a little bit. We had a bit of an outline when we discussed about epidermal variable sensors on day 1. We'll explore the different sensing platforms that we have. What are the different types of sensing platforms for sweat-based epidermal devices? What materials could be used for building those devices? And we're going to be looking at how signal conditioning and power strategies can help us improve the performance of the sensors and review some of the clinical and field applications that have been performed with these epidermal variable sensors. Okay, first of all, let's just do a quick recap as to why sweat is an important biofluid. Why is it important? What are the advantages of sweat as a biofluid? Well, the first most important thing is it's non-invasive, right? You don't need to you don't need any sampling device to collect sweat, in the sense that you sweat naturally, right? The problem is when you can't sweat, right? So I think most of the people who work on sweat-based sensors, they also kind of employ methods in which sweat can be generated. But again, as I said, the advantage of sweat as a biofluid is the most important thing is it can be accessed in a non-invasive manner, and it's readily available as well. What does it have? It contains most of the metabolites because it's all coming through your sweat glands. It contains glucose, it contains lactate, it contains cortisol, it contains electrolytes. Easily accessible, has these analytes. Is that all that we need? One more thing we need is there should be a good correlation with blood. That is a big question mark here. It correlates with blood for select analytes. It does not correlate for all the analytes. That's the problem we have with sweat as biofluid. I think that's why sweat-based glucose monitoring is not very common, because there's not a good correlation of the concentration of glucose in sweat when you compare that to that in venous blood, right? While in skin interstitial fluid you have a much better correlation when you compare the concentration of glucose in skin ISF versus venous blood. So the challenges that we have when we talk about epidermal microfluidics or epidermal variable devices is, number one, the sweat rate will vary, and the other thing you can have is you can have contamination with something that's already on the surface, right? A lot of you use deodorants and stuff like that. Imagine you try to collect sweat and then you're spraying your own deodorant, and you have a sensor which is meant to be monitoring alcohol. Yeah, you've already contaminated your surface there, making it really hard, right? So this is thing. So if you look at the composition of sweat on your screen, I'm sorry for the small fonts, it's mainly made up of water. It has some sodium, some potassium, and also got some urea there. And I think I showed you some examples wherein the variable epidermal sensors were used for monitoring urea in sweat as well. Okay, sweat as a biofluid, there's been a lot of trend towards measuring urea in sweat, and there have been the different methods that have been used for this. People have used amperometric methods, people have used impedimetric methods, potentiometric methods, voltammetric methods, piezoelectrometric methods, Raman spectroscopy, which is again an optical method, fluorescence spectroscopy, electrochemical luminescence, and colorimetry. So different methods here, and you can see most of this has been done. In fact, even this one is your electrochemical. So most of it is done using electrochemical methods. You have this mass-based, which is your piezoelectrometric method, and the optical here is just this little quadrant that you see here. Okay, so again, it brings us back to the same question: why are electrochemical sensors more popular even when it comes to epidermal wearable devices? And the answer to that is portability, ease of miniaturization, right? That explains this large fraction of this large pie of this whole thing being more electrochemical sensor based compared to the optical here and the mass based. Piezo is really hard. This has actually been pulled out of a paper which is on sweat analysis for urea sensing, which is in a paper published in 2023 in Analytical Methods. This paper should be also be made available to you through the folder that will be made available by Professor Khalid, or it's already been made available. Okay. Who are the pioneers who work in the area of sweat? I think we've got 3 main people who work here. One of them is a very young guy, Wei Gao. Some of you idolize him, I know that. A young guy, he's just started doing it, working in this area of, he's from Caltech in Pasadena in the United States. Actually, all the 3 of them are from the United States. You know, you may ask the question, why are all these people from the United States and why do we not have anybody from Asia or from Europe? And the answer to that is there's a lot of funding that's available for adventurous research which are kind of, you may have, which are high risk research projects, and those are normally funded by US funding agencies more than you would find in Europe or in India. In Europe and India, they like to have projects which have got minimum risks, something that's been tried and tested, something where you have some proof of concept studies or valid data there, and you don't get anything to do kind of anything under the blue sky kind of research there in Europe and in Asia as well. The main big player in this area is Professor Joe Wang. Joe Wang is the book that I recommended in the last lecture was that on analytical methods or analytical electrochemical methods was by Joe Wang. He's the head of the Center for Wearable Sensors. He's a distinguished professor of nanoengineering in the University of California in San Diego. He's also a director of the Center of Wearable Sensors, co-director of Center of Mobile Health Solutions or Systems, fellow of the Royal Society of Chemistry, fellow of the Electrochemical Society, and he works in this Department of Chemical and Nanoengineering in the University of California in San Diego. A fantastic person. I've heard some of his talks at Imperial and at various World Congress on Biosensing conference platforms as well. Another person who works in this area of epidermal wearable devices is Professor John Rogers. John Rogers is based in the Northwestern University in the Department of Material Science and Engineering in the United States. And again, his area or his expertise is mainly on soft electronic and microfluidic systems for skin. So he works in this area which is popularly known as epidermal microfluidics, right? This is where you try to address the problem of variability in sweat. So rather than just monitoring sweat in real time, you collect them using a microfluidic system and then you analyze them, right? You don't have to actually, when you talk about wearable devices, you don't have to measure every analyte in real time every second, right? Even the best case example that you have, that is the commercially available continuous glucose monitoring sensors, they measure glucose and give you values. They measure glucose, they have the capability of measuring every second or doing multiple measurements every second. But what they normally do is they measure and give you an output value. That value changes every 15 minutes. Okay, so these are the pioneers that you have here who are working in this area. So if anybody wants to work in these areas of epidermal variables, or even for that matter subdermal variables. In fact, I know Professor Joe Wang has also published some papers on microneedle based subdermal variable sensors as well. So it's to keep on looking at which biological matrix for what would be more beneficial, and that's how they make a call as to what to work on. This is Wei Gao, a very young chap here. You can see his very flexible kind of sensors here. You can see it's on a flexible material. You can see this, those lines there. Those are the basically showing you, and these are some adhesives to stick onto the skin surface. And you have these. This is your working electrode. This is your outside. This is your counter, and then you have your reference electrodes as well. And I think they've got multiple working electrodes in this particular setup here. So the different sensing platforms that you have for sweat are tattoo based sensors. People have made tattoos, made up of gold nanoparticles of graphene PEDOT-PSS, which is basically, this is polystyrene sulfonate, and the other one is a thiophene here, polyethylene dioxylate thiophene. And then people have also used microfluidic patches. As I mentioned, I'll show you some of those microfluidic patches, very thin microfluidic patches, which basically have got reservoirs to collect the sweat and then to do the analysis. Another one which is very popular in India, there are many people in India who are working in this area, is paper based microfluidic systems. One of my previous colleagues, Amit Asthana, who's in Hyderabad, is working a lot on paper based devices, and I know that Bill and Melinda Gates Foundation have funded a lot of projects in India which is based on basically paper-based microfluidic devices, or paper-based devices for point of care and wearable devices. The pioneer for this, of course, was George Whitesides from the US again, and I think he's got some projects running wherein the projects have been funded by Bill Gates and Melinda Gates Foundation. We're also going to be looking at different platforms, bandage style flexible electronics, where you have like a bandage and that's got flexible electronics. Has anybody heard about dissolvable electronics? So the problem with electronics is that electronics, they generate a lot of waste. I mean, you might have seen from the pandemic where everybody was having those laminar flow devices, and some of them were having electronic units. They had the PCBs which had got you could easily see the capacitors and the little screens and stuff like that. Even the people who use pregnancy tests at home, or people who use glucomonitors, blood glucomonitors, there you just have the strips that are being generated. The rest of the electronics are being reused, but there are a lot of laminar flow devices which come with the electronics, and the biggest problem there is the electronic waste that you have. That's why point of care devices are kind of going, people are looking for alternate ways, especially methods which are in accordance to green chemistry. You can look at dissolvable circuits, dissolvable electrical components. People are looking at natural material based devices, silk, paper. PDMS is not natural, PDMS is hard to dispose of, it's an elastomer. So there are these different materials that are being used for these bandage style flexible electronics as well. And then there are these fingertip passive sweat sensors, wherein you take the sweat and you transfer it to a finger. I showed an example of this yesterday. For some funny reason, I also have seen this kind of thing wherein you have the microneedle and sweat based hybrid systems, something which is measuring under the skin and on top of the skin as well. So this is like more like a hybrid system here. So the microneedle based sweat hybrid system that you have here. Okay, let's start looking at some of the examples here, and then we'll have 4 specific examples, but let's have a look at this one here, which is what we call as an OECT, which is an organic electrochemical transistor device. Yesterday the transistor device was explained by Professor Khaled. He mentioned about having a gate, a source and a drain, right? And the gate part is the part where your solution, normally you have an extended gate, that's where your solution normally sits, and then you have a source and a drain, and you normally apply the potential, or you either monitor changes in the potential, or you keep the conducting channel, the conducting current, the conductance fixed, and you measure the changes in the potential there. So it's a potentiometric method for monitoring stuff. So that's a transistor here, and the other rest OEC stands for organic electrochemical transistor here. So this is basically a highly sensitive biosensing device that has been produced based on PEDOT-PSS, which I mentioned is nothing but a thiophylline and polystyrene sulfonate based organic electrochemical transistor. You can see that the limit of detection you're getting it is in micromoles, which is fantastic. How you make these devices, again, you're having a flexible material here because you're talking about your sweat side of things. You need to create channels so that you can collect the sweat. So they're using laser induced micro patterning. So you can draw the lines with laser, you can actually ablate and you can create channels. So they use this labor induced micro patterning to get these kind of channels on the PEDOT structures there. And then basically this is a single use epidermal sensor here. You can see there's a reservoir here, and on the back of it is your PEDOT layer, which is basically an organic electrochemically active layer. And this was published in the ACS sensors there, and you're getting good limits of detection here, which is in the micromolar range in this particular case here, right? So this is a nice paper which was published in ACS Applied Materials in 2024. It's one of the recent papers that have just come out. All right, and then I'm going to talk a little bit about epidermal microfluidics. You can see the man himself, John Rogers, with some of his device, especially in the form of tattoos that have been stuck on his face. We've got some nice little tattoos that he's got there, and he's also holding a PDMS device there. And this is a nice little device which is sitting on your forearm. What it does is it's basically again got a series of, so it's got different multiple layers. So you have the sensing layer which should be on the top, and you have the bottom layer is more like a microfluidic layer for collecting the sweat, and you collect the sweat, and then you have 4 different reservoirs here. You can see the different colors there, and they're measuring things in a colorimetric manner. That's a nice thing about epidermal microfluidics. Now colorimetry clearly is not an electrochemical method, it's more like a visual method, it's more like an optical method there where you see a change in color. Very, very interesting, and I can give you examples. When we had this whole incidence of animal influenza, birds were being culled in the UK, and so we were asked to design projects which would allow us to ensure that the birds are not culled or killed just under thinking that you don't need to wipe off the entire flock that you had on a farm there. So people wanted to have visual, for farmers, what's the easiest way to develop test for farmers? Something that's visual, right? A color change. So we had these devices which were based on APTA sensors, and they were more for visual ones. So what you would do is take a swab of the backside of the bird and then dip it into an Eppendorf tube, and then which would have a solution, and then you would see a color change. So that's how you would transfer the biomarkers from the backside, which is basically of the bird, and transfer it into the Eppendorf tubes, and then you could see the color change. That's a nice way of doing colorimetry. Is a nice way of it's a simple way. It gives you qualitative information. It tells you yes or no, but it doesn't tell you about the quantitative information. It doesn't tell you how much, right? So this is a nice colorimetry thing which tells you how much basically of this hydration is there, how much of urea and stuff like that are there, but it doesn't tell you, it tells you if it's there or not, but it doesn't tell you about how much of it is there. And then this is basically another epidermal microfluidic device which also has a pump. This is so you have the pumps which are designed on top of the it's like more your pressure pump. You just apply a thumb pressure and you push the device so that it gets distributed into these channels that you have here. So you have your sweat that's collected here, you apply the pump which is designed on top of this polymeric layer. It's the pump inlet and pump outlet. You apply pressure and it then pushes everything inwards towards these radial channels here, and you can see what's happening here is it starts to fill up all the different channels, and you have the biosensors underneath the channels which can do multi-analyte sensing as well. So this is a nice interesting paper, again published in one of the journals that we all dream in publishing, Nature Communications. This was published back in 2019, again by John Rogers. This is an example of where iontophoresis is being used. So we spoke about the gluco watch yesterday. So basically what you do is you apply a current. When you apply a current, then it kind of extracts it makes the ionic flow in the direction so that it comes out of the skin, and then you induce sweat there, and then that's how you normally measure this in using the ionophoric measure through iontophoresis and measuring the current. You can get your epidermal variables as well. So the gluco watch falls into the category of the gluco watch thing that you said, and this is some work that has been pioneered by Pat Connolly from Strathclyde University. If you want to look into this work, you can easily follow her up on that particular website there. It's nice, it's very nice in the sense that, again, you don't need to extract anything, you don't need to push anything under the skin, there's no needles there. It's a very neat way of conducting things, but again, limitation, there's a limited number of analytes that can be measured using epidermal variables. So what are the materials that have been used for your epidermal sensors? Well, the people have used things like PET, PI, which is polyamide, PDMS, EcoFlex. These are all very flexible materials. You can actually make sheets, you can make rolls. If you can make rolls of anything and then cut them down into something that can be easily wrapped around your finger or wrapped around your wrist or wrapped around your arm, makes life much more easier. Anything that can be made in rolls is a scalable technology. You can make multiple rolls, and it's low cost as well, right? So one of the good things with epidermal wearable devices and the technologies that are used for making these epidermal devices are those technologies are scalable devices, because you use substrates that can easily be kind of formed into rolls. And then on top of that, you print your layer or your conducting material, and normally it's not as if you're using real gold. It's just 50 nanometers, 100 nanometers of layers of gold silver, carbon nanotubes or graphene that can be printed on top of this rolling material. And it's, you can imagine, and in some cases what they use is just a glorified inkjet printer. So you take inkjet cartridges, replace them with cartridges which can give you gold ink. You can get gold in the form of ink, you can get silver in the form of ink, you can get carbon nanotubes and graphene in the form of inks. So you can replace the cartridges with those ink cartridges made up of gold, silver, carbon nanotubes and graphene, and you can print your own devices. So you can literally, if you get the roll, if you order the roll on Amazon or this thing, and if you have a printer cartridge which you can modify, you can make them in your own home settings. Okay, and then you can use these to then, and then you can, on top of that, if you're trying to monitor ions, then you need an ion selective membrane. If you're doing enzymes, then you can also get enzymatic inks as well. You can mix the enzymatic inks in your conducting inks, and you can have something wherein they are nicely, you can form thin layers there. And you also have this whole option of printed electronics. This is what I just mentioned about. You can have screen printed electrodes, you can have inkjet printer based electrodes, and you can also have aerosol jets wherein you can just spray them and then dry them. Somebody was talking about electrospinning as well. You can also use electrospinning to electrospin on top of your substrate material. This is another example that we have on wearable sensors which has been made of different materials. This probably work is come from, again, Wei Gao, right? This is basically published in Nature. I sometimes it's a mystery how a person can publish so many papers all in Nature. That just shows the quality of the work. It's Nature around these. So people who know the field of publishing, you have journals which have got very high impact factor journal. So all the Nature journals, the science journals, impact factor is good, but it's not the only thing, right? But for somebody to publish continuously in journals of especially Nature thing, some of it is reputation and most of it is the good quality of work that comes with it as well. Okay, so this is where he's basically shown how he's used a fully integrated wearable sensing platform using a PET device and substrate. Sorry, this picture basically is to show you wearable sweat sensors made of different materials. This one has come from the group of Wei Gao. The second one is where you have embroidered, so you can use fibers, with conducting fibers. You can use a sewing machine, and you can have a sewing machine and you can make your own electrodes on fibers as well. That is a nice and neat way of making wearable devices. This is, I think, probably the work of done by Ferhat Güder from Imperial College again. Again, very, very neat. You can see here he's printed the counter electrode, the working electrode, and there's a reference electrode here which is not very, very clean, nearly visible. And this has been printed on a nice cotton t-shirt as well, right? And then these are some examples of sensor patches for monitoring sweat. What they've done is it's basically a band-aid pad on which you have your sensor which has been printed there outside and inside. You can see here, this is the inside one. Somebody was talking about wound healing. So if you want to monitor wound healing, this is a nice way of having sensors which can have iridium oxide sensors to look at the low pH or the pH dropping down, and they can also have sensors which can measure glucose as well. I think somebody was looking for wound healing. Yeah, that's wound healing. So you can, this kind of a thing will be very useful here. The references are here on this slide here, so you can pull out these references once you get the slides. Yeah, so it shouldn't be a problem here. And then you have this example of this sweat analysis thing, which is based on a foldable all paper substrate. Again, this is an example where paper has been used as a substrate, and you have this sweat analysis design there. And then coming to this example of silk, I mentioned of silk to a few of you yesterday, using silk based carbon materials. This is your silk based sensors which have been used as a wearable electrochemical sensors as a working electrode. That's the example of the silk one. And the last one is basically a wearable laser. This is a laser engraved sensor which is again used for sweat detection for different analytes. I think the most common one is urea. I think urea is used a lot for sweat detection because it's got its own challenges, and I think there's a good correlation of urea in sweat and urea in blood as well. Transduction mechanisms similar to what we saw this morning and yesterday. As I said, the basic principles that we've seen, potentiometric methods are used for ion sensitive electrodes. Of course, as the name indicates, ions, meaning that you're looking at sodium and potassium ions, right? For these you normally have those little membrane like structures which are very selective to the different ions. They're more like size exclusion membranes. They are sensitive to the differences in size, and they are able to differentiate between sodium ions and potassium ions based on just the size. right? So those are membranes that are used in ion selective electrodes. And then amperometric sensors. I mean, I think you must be bored now with listening to glucose sensors, but that's how it works. Glucose sensors and lactate sensors, which are based on enzymes. The same transduction mechanisms are used in epidermal wearable devices. In terms of impedance, you can measure hydration. Most of you might be having a weighing scale at home which measures bioimpedance. Anybody come across that? So if you can buy these off Amazon these days. You have a weighing scale which will also tell you about your BMI. How does it do that? It can calculate the total water in your body and calculate your weight, calculate your dry weight, and it can calculate your muscle weight, calculate your bone weight. So it does some impedance measurements. It's called bioimpedance measurements. You step on the weighing scale and it does a lot of calculations, and then it tells you, okay, your BMI is this much based on your weight, your height, and the dry weight in your body, your muscle weight and stuff like that, and gives you your BMI, right? Has anybody tried that? Has anybody used that in home settings? No? You're all very young. Wait till you get to past your 40s. I think everybody will start using them. Colorimetry. Colorimetry is very, very useful. I've already given you examples wherein it's a nice way of visual detection, point of care. If you see a color change, it should be a significant color change, ideally from blue to red, green to red, and things like that are very much visual, although it might be an impairment for people who are suffering from color blindness. Okay, the instrumentation for this will be introduced by Professor Khalid. Just to let you know what are the different kinds of instrumentations that these people used, because you might have seen some of those pictures where they have those very thin layers on which they have the different forms, and I showed you some instrumentation when I introduced you to the slide on biosensors. You have some instrumentation at the front end which will involve things like filters, amplifiers, and ADC units, analog digital converting units. And then you will also have some microcontrollers. You're getting data coming out of it. You don't want the person to be collecting the data and then processing the data. So you need to have a microcontroller which can do all the processing for you, and the processing unit can then smooth the data. You can have your machine learning and AI codes on it which can then help in making more sense of the data, and it can predict a value. Yeah, you can get a predicted value based on all the microcontrollers. So you can have data handling there, and then you can have all of this BLE and NFC, which Khalid will be able to explain you much better when he has his lecture on the instrumentation side of things. In terms of powers, you normally could have a battery there. It could be a tiny little lithium battery there, or you could have, we have also shown you examples wherein people have started integrating biofuel cells, lactate based biofuel cells. You can also integrate that, and you can make these into a fully integrated sensors there. And then how do you collect the data, right? The data will be normally accessed through the cloud. You can have a smartphone, or you can have cloud based analytics. You can have what we call as an iCloud, wherein you have everything goes into the iCloud, and from the iCloud it can be relayed onto your smartphone using a Wi-Fi signal or things like that. So these are the standard parts of a wearable sensor that you need to this thing. Main focus of this whole Ligyan series is to kind of make you aware of these different components that go in to build a wearable sensor. And then looking into applications in health and fitness, they have been used for different forms of applications in health and fitness, right? I mean, most of these kind of sensors, wearable sensors, are used by fitness freaks, or I mean, now these days there's a big I would say the people are becoming more and more alert. People are using sensors for very different reasons. People who are, I mean, it's all started off by trying to address a problem. The problem was diabetes. It all started off by addressing a problem of type 1 diabetes, right? When you try to look into type 1 diabetes, the problem there is it's more in juveniles, more in kids. So you needed a carer there. So you needed something that could give you continuous reading, and then something that could give you could take the control of you, and then decide for you if you needed an insulin shot or not, if you needed to adjust insulin dosage or not. That was meant for type 1 diabetes, right? That's how the whole concept of artificial pancreas came in. That's something that again Professor Khalid will be talking about, right? So you can have that, that was the main application, and then it moved into type 2 diabetes. People want to see how the profile looks like over the days, right? Type 2 diabetes is more lifestyle diabetes. This is something for which we ourselves are responsible, right? You can have insulin resistance. You can have, you can basically have diabetes because of very sedentary lifestyles. I mean, I've been sitting on this chair for the last 3 hours, not getting up at all, right? And I would have preferred doing this lecture walking around. So I know if I measure my glucose now, the numbers wouldn't look very good, right? So type 2 diabetes is more lifestyle related diabetes, more sedentary diabetes there. So now the whole application of these, especially for these wearables, is more towards well-being, and well-being includes measuring your metabolites like glucose and lactate. People are running around and they're checking the lactate levels, people are checking their glucose levels, people are checking to see how long are the glucose levels staying within the normal zone and how long are they staying outside the zone. So people are becoming much more aware of this health problem and making sure that they're looking after their own health care. So these wearable devices are actually helping you decentralize healthcare from big hospital settings to a GP practice, now in your more home care settings there. Okay, and as I said, so these wearable devices are finding adjunctive glucose monitoring for diabetes, cortisol for stress and circadian rhythms, and as I said, urea, one of the biggest biomarkers that are normally explored by the epidermal wearable sensors, which is a bioindicator or a biomarker of chronic kidney function disorder. That's where you measure urea there as well. What I'm going to do in the next part is run you past 4 different papers that have been published. One was published in 2018 by Hong et al., which is basically on using epidermal devices for looking at glucose and the vital signs patches. And then Sempionato, I think this is Joseph Wang's group, which published in 2021, fingertip sweat sensor. And then Mishra published the kidney biomarker, again, which is a glorified name for calling urea biosensor. And then we had Ian et al., which is an example of how lactate was monitored, and they also used lactate was monitored. Lactate powered biofuel cells were employed for this particular study here. Okay, so this is the first paper. This is by a group from, I believe, which is from South Korea. Looking at the names, Hong was the first author here, and they developed this biosensor, which is on multifunctional wearable systems that are integrating sweat based sensing and vital sign monitoring to estimate pre and post exercise glucose levels. So clear cut, simple thing wherein you have a wearable device for monitoring glucose before the exercise and after the exercise, just to look at the device. So you're having a disposable device here, you use it, once you've used it you can then dispose it off as well. But it's good to, again, this is continuous monitoring, but this is continuous monitoring over a certain duration. It's not like 14 days continuous monitoring, it's monitoring over a certain period of time which is pre-exercise and post-exercise. That's it, just monitoring those peaks just to see how much does your sugar level actually spike, right? That's your case study. That's the first case study, which is done by Hong et al., and this paper is available for you through the organizers. And then there's a second paper which was published in 2021 in Nature Biomedical Engineering, another journal that all of us aspire to publish into. This is by Joe Wang's group, and this is about an epidermal patch which is used for simultaneous monitoring of hemodynamic and metabolic biomarkers. A very nice way of saying, when I say metabolic biomarkers, I'm mainly talking about glucose, lactate, again urea, ketones, and sometimes they have also used for monitoring alcohol as well. So you can see they're looking at consumption of glucose via glycolysis, and they're looking at oxygen depletion and lactate generation as well. This is a good continuous and simultaneous acoustic and electrochemical sensing. So they're also doing using the acoustic sensors there as well. Those are more physical sensors, right? Surface acoustic waves. They use more for acoustic sensing, and there are integrated wearable devices which should enrich and understand the body's response to daily activities, again for prediction of abnormal physiological changes, right? As we age, we want to look at how the physiology is progressing. Our metabolism slows down, so it's nice to have devices that can give us an indication about our metabolism. This then helps us to make changes in our lifestyles as well. You know, more exercise, less food, and things like that. This is all possible using epidermal microfluidic patches, epidermal wearable devices. This one is an example which I've already shown. I mean, I like this a lot. This is by Mishra et al., and this was published in ACS Sensors. This is again another good journal. It's got an impact factor of more than 10. It's a nice journal, and it's relatively new compared to the other ones. and this is where they've used colorimetry here. You can see you've got the circle here, and then you've got those 4 different patches. So you're actually getting n equals 4 readings, and you've got 4 different dots you can see which are arranged radially, and this is your sample collection point, and then this is how you activate the pump. So you've got the 4 pumps on 1, 2, 3, 4. These are the 4 pumps. You collect the sweat under this particular point, and then you push the pump, and that pump then makes the fluid to flow in this 4 different channels and be collected in the radial designs there. And you can do multi-analyte sensing of 4 different analytes, and not only are you doing of the 4 different analytes, you're also doing it 4 times. That also helps you confirm, and that also shows how reproducible the tests are. I know it's epidermal microfluidics, epidermal variables, so some of you might be falling asleep. I don't blame you for that. Okay, and also we've had long sessions before this as well. Okay, and this is the 4th one here. This is a study that was done by Wei Gao, and this was published in Science Robotics. Nature and Science happen to be the most popular magazines, very popular journals. We've got very high impact factor journals, and what he has done here is, instead of using an external power source, he's actually used a biofuel powered source which runs on a lactate sensor. So it uses the lactate in the body to power the whole series, the power the circuit, and he's used that for doing multiplexed and wireless biosensing at human machine interfaces as well. So another very good example of how epidermal wearable devices can be used, this time generating its own power to power its own biosensing device there. So to contextualize these, what are we doing in our lab? We call our lab as SSRG. It's got nothing to do with my name. It's Smart Sensors Research Group. We work more on subdermal wearable devices, and the subdermal microneedle sensors are mainly meant to target interstitial fluid, not sweat. So that's how we're doing it different. We are also using AI enhanced. So if you look at some of the work that has been done in these papers, they don't mention a lot about the machine learning and AI, but they are. Wei Gao is also using AI, machine learning and AI. Joe Wang is also using that. We normally use that for artifact correction and for continuous data, and we've also used ours in testing in humans and ambulatory livestock as well. We don't claim to be better than the epidermal variables, especially if you're talking about glucose sensing. We say the sweat sensors, we can use them as complementary methods for multimodal testing as well. So if you look at the image I showed you here, you've got the circle there with 4 different sensors there. In our case, we use microneedles which have got different islands with 4 different arrays of microneedles, just to kind of put things into the context there. So again, we can use it in, and that's what we call as using it in multimodal diagnostics as well. Multimodal in the sense that you can also have a electrochemical mode of detection, and you can also have a colorimetric mode of detection there as well. Okay, so coming to the future outlook and challenges, well, I think if you want to work on epidermal wearable sensors, the things that you have to mainly focus on are how you can standardize sweat plasma correlations. I mean, that's going to be a big problem, because even people who are working on epidermal sensors, you have to make the person sweat. It's not always possible to make a person sweat unless you have getting the person in ambient conditions, even room conditions. You can't do that because you have the AC running. I think everybody is hardly sweating here, but as soon as we step out, we start to sweat, right? But then clearly we'll get different values sitting inside the room and outside the room. So we need to have a good correlation study where you can look at the amount of sweat being generated in response to the changes in temperature there as well. So we need some standardization there, which will allow us to then correlate the sweat with the plasma as well. Sweat stimulation methods. How do you stimulate sweat? I mean, you can make a person either exercise, or the other thing is you can use iontophoresis. We saw how a glucowatch was using iontophoresis to extract the sweat out and get the sensors to and get the sweat out and be able to do measurements in the sweat there. And then we also we've also seen how we have looking at the Wei Gao paper there, where he was using a lactate based biofuel cell. You can improve the power autonomy. When you say power autonomy, meaning that you're trying to have a self powered device which can then power its own biosensing unit, because you need to apply voltage. You need power sources for that, and this can allow in improving power autonomy, and also most important is wireless range. And the other important thing is data. What do you do with the data? How many of you are happy for your data to be uploaded onto into an iCloud which doesn't have any security arrangements, right? Nobody would want. I don't want you to know what my heart rate is right now. How many of you are, no matter how young you are, unless you want to boast about your heart rate being very low, even when you're exercising, you don't want to display, you don't want to share your heart rate. So this is all personal data, and this is a very important problem that we need to understand. So sometimes when you apply for grant funding, they ask you, how are you going to store your data? What about data protection? How are you going to, what are the regulations around your data protection there? How much of this data is going to be in the public domain? People would not want it to be in the public domain. I showed you images of doing clinical studies, and I covered the image of the participant. The reason there being, the participants do not want to be identified. Sometimes you see images of babies being displayed. That's completely unethical. If you are showing images of babies, you have to cover the faces, right? So how do you improve the power autonomy, and how do you improve the wireless range? And to this I would also add about data security as well. Right? How do we make sure that the data is secure, and then add on to this comfort, biocompatibility, and the user compliance as well. I think that's probably my last slide, and we can now open the floor for further discussions on what are the pros and cons of sweat versus interstitial fluid monitoring, what material innovations enable flexible biosensors, and what are the barriers to adoption in clinical settings. We can have this discussion on these three important points. I mean, I have put my case forward. I think epidermal wearable devices are fantastic, right? If I would not introduce you to both epidermal and subdermal wearable devices, I wouldn't be doing justice to you, because I want you to understand both these fields, both these areas, and then choose the pros and cons, and then make your own choices. As I said, there's no competition going on between these different types of wearable devices. They are complementary to each other. Some are giving you good information. If you have a device which is just in the form of a patch which is sticking to your skin and giving you 4 readings in a colorimetric manner, and you can quickly see whether your urea levels are high or not, you wouldn't mind that. I think it depends on your requirement, right? As I said, classic example, pregnancy test kits. The previous versions of pregnancy test kits were ones which would give you a yes or a no, answer, right? The new ones are the ones which tell you to what extent, right? How many weeks as well, right? So it all depends on what is the level of information you require, right? I think with that I'll open the floor for discussions. If you have any questions, we can have a chat about the pros and cons of sweat versus ISF monitoring. I mean, based on what you've already been presented, how many of you would actually venture, if you had a choice between epidermal and subdermal, how many of you would venture into subdermal, and how many of you would venture into epidermal? Let's do a show of hands for epidermal.
Yes, epidermal devices. How many of you prefer?
Very easy to make. If you're doing a PhD on it, I think this will be an easy ride, because everything gets much more easier. Your research ethics approvals gets much more easier. You're not penetrating the skin. You're sitting on the surface of the skin. You can get some information there, and you can use it in both multimodal manner. You can use it for electrochemical and optical sensing. How many of you would go for a PhD on epidermal wearables?
We can have a show of hands. No worries. Do you understand epidermal sensing, right? So what sort of analytes can we monitor in epidermal devices?
ग्लूकोज स्वेट से और लैक्टेट जो बताया था.
Okay.
Sir, epidermal wearables can be used for a wide range of age group peoples.
Okay.
Because for babies, the small children, like epidermal can be a good option rather than the subdermal.
Yes, absolutely. I mean, for babies, epidermal sensors, if they prove, it will mainly depend on the function. But if you're trying to monitor urea in babies, perfect. Just stick a patch on the baby, right? You don't need, there's no need for the needles that, I mean, have you ever seen a baby in a neonate unit? Oh, it's a painful scenario. I mean, I wanted to become a pediatrician when I was growing up, but after I joined Liverpool, I had a chance to go to the Alder Hey Children's Hospital, and I looked at the babies there in the neonatal unit. not a pleasing scenario. You know, babies look cute when they're smiling and but if you see babies crying, it's not an ideal case scenario, or babies in incubators. Yeah, so it's a good example you've given. Yeah, so you're absolutely right there, that if you would easily get a research ethics approvals if your epidermal wearable device is capable of giving you the information for a baby, because this epidermal could be classified as completely non-invasive, right? You're not doing any damage to the skin. Subdermal would be minimally invasive.
Okay, so we can go point by point. So what do you think are the, from your side, from the audience side, what are the pros? We can list pros of sweat monitoring. So from your understanding, you can tell us. So
what do you think
are the advantages
of sweat?
It is easily available.
Yes.
It is easily
Well
available.
done. Good answer. Easily accessible.
Yes.
Right. You can generate sweat easily. If you want somebody to sweat, just increase the temperature of the surrounding, and you can get the person to sweat. Right. And then if you want to collect it, you can use a microfluidic device, a very, very basic microfluidic device. Even a paper. You can even use a Wattman filter paper to collect sweat. It's like collecting tears. There's a strip called a Schwimmer's tears, because you get very small volumes there. Right. That's good. That's the advantage of sweat. What else?
So as a target like analyte fluid, it's easier to collect it. Yeah. Anything else?
Sir, actually the
Sit. Don't worry. Yeah.
the sweat is readily available in the human body.
Yes.
Already there, and also we can use in the region like India's, because it is near the equatorial line, so there the temperature is high, so the the person has the more sweat concentration rather than the region in the western countries, so it is more useful for us also.
I agree, but just to make a point very clear, I think the sale of deodorants is not very less in the western countries than in India. People there also sweat quite a lot. It's just that it's not very evident.
And also the cons is basically the concentration of the glucose.
Pardon?
The problems of the sweat uses analytics, because the concentration of the glucose is not prominent, or it is variable in the sweat, so we cannot use.
Yeah, I agree with you on that.
So
I'll take that point as well. But what is the bigger problem there? There's no good correlation. Exactly.
Yes, there is no good correlation. Yes.
Good. I think we can move to the ISF now.
Just coming to this. Yeah. Just
Yes,
one minute.
please.
The quantity of sweat can be also an issue, because some people sweat a lot, and some
Agreed.
are also having a genetic condition where they excessively sweat,
Yes.
but others have different.
Yes. People who have problems with their hypothalamus in the brain, they have this condition wherein basically they sweat profusely. Just eating makes them sweat. Just sitting there quietly makes them sweat. Have they switched off the AC, or are we testing the sweat-based sensors? I thought somebody's trying to make me sweat now.
Sir, also another problem is like in the winter season the person does not sweat a lot, and in the summer it
Yes,
it
agree.
is time dependent also.
Yeah.
Okay, what I also wanted to mention is regarding the correlation. Okay, it's not very strong that it justifies, let's say, glucose. The correlation between blood glucose and sweat glucose is not strong enough to justify a diagnostic application. But there are certain applications where you just want to get an idea.
Yeah.
It's
Point measurements.
point measurements.
Agree.
It
So
can be
more for point of care kind of things rather than for continuous monitoring or for having it as a wearable device. Because wearable devices, as we say, something that you wear for a particular duration, and that duration could be anything ranging from 4 hours to 6 hours to a whole day, or so I think that we'll do that. What I'll do is at the end of this session, I'll show you a continuous glucose monitoring sensor. I'll implant it on myself,
Okay.
and then we can just kind of get some idea there as well, and then it's going to sit there for 14 days. But I'll send you a picture, which you can circulate to show what happens when you remove it outside. Show you a little bit of blood there as well, to show you exactly how deep it goes in.
Yeah, we can do that. So we have some continuous glucose monitoring sensors, and we also have microneedles. We have PCB based sensors that we can show you now in the next session, as well as in the during the hands-on experimental.
Yeah.
So compared to sweat, now like for example, I guess has anybody done a comprehensive study of what sort of analytes that can be monitored in sweat? So in ISF, of course, it's the so in ISF we find most of the analytes that are present in plasma. We are able to detect in ISF as well. What about sweat? So for example, in ISF, the main issue is the size of the molecule. The smaller the size, the more available they are in the ISF. Is it the same in sweat, or how does it? What is the link?
Sweat is normally coming through the sweat glands.
Right.
But having said, so you expect to see metabolites there. That's why urea is very common, glucose is very common, lactate is very common, alcohol is very common.
In sweat.
I mean, you guys are this thing. So when people who are excessive drinkers, even their sweat stinks of alcohol. You can actually, if you monitor their sweat, you can even get alcohol from their sweat as well. So because that's a very small molecule, ethanol, C2H5OH is very small. You can easily calculate. You can see it there as well. You can see ketones in sweat as well.
In sweat. Okay. Interesting. Any other points you would like to make?
Again, one more thing I'd like to mention is this: when you're close to the sweat glands, you also have certain hormones being hormonal ducts as well. Cortisol. But the only problem there is that there's no good correlation between these cortisol and your the hormones that's produced, even the reproductive hormones, and blood. That's why people are not using that for those kind of studies.
So we may not be able to use it for like therapeutic or diagnostic applications.
No, it's more for well-being kind of.
Yeah, it's kind of well-being, digital wellness, or kind of wellness where we want to get a rough idea of health.
Yes.
Without going too invasive, we can still have some idea from the sweat monitoring device. So again, in ISF, I guess ISF is a very good fluid to use for monitoring applications, for continuous monitoring applications, I guess. So so in terms of so that is a pro for ISF, that it is having strong correlation with blood analyte levels, so you can use it for many applications. So if you are developing something for a diagnostic or therapeutic application, then ISF would be a better target.
Absolutely.
And if you are doing something just to get an idea, or sort of which has some wellness application without any direct health related application, then we could go for sweat, I suppose. So moving on, so many people have developed these flexible biosensors. So what would you say is the challenge remaining to be overcome in flexible biosensors?
I think what we need there is, so what's happening is we are still sticking to polymeric materials. I think we should start looking into natural polymeric materials.
Which are biodegradable.
I think that green, somebody mentioned green chemistry yesterday. I can't remember, it was a gentleman who mentioned about green chemistry, spoke about green chemistry. So if you're talking about green chemistry, because I mean we are actually going through a lot of this pollution thing that's going on around. So the AC started again. I think as soon as we move to ISF, they've stopped making me sweat now. So basically, I think it's all about biocompatibility, and it's all about the environmental side of things as well.
Okay. So do you have any so what are the typical polymers that people are currently using? So polyamide is one,
PDOT, PSS, and all these are all materials that are being used. PSS, polyamide, PET, Ecoflex, these are the materials that are being used right now. At the moment, people are not worried too much about the environmental side of things, because this is research in early days. It's only the concern will be when you start getting multiple of these devices. To wrap them, you need, so I think silk would be a fantastic polymer, because it's a natural polymer. Cotton, paper, anything that's natural polymer, and then inks that you use for them.
Okay. Nice. Now it's good to know. So and Okay, moving on. So, what are the barriers to adoption in clinical settings? What would you say, doctors, from your experience?
I mean, if I think the gentleman there gave a perfect example on babies. So neonates. I mean, nobody wants to see already these, you have so many things being already kind of plumbed into the bodies of these neonates. The last thing you want to see is another device which is again a bit minimally invasive or invasive to go into these neonates. But if you can have something that can be stuck to the diapers, so that you can monitor from sweat, or we can monitor in urine and stuff like that. That's perfect. So the barriers to adoption is epidermal microfluidic devices or epidermal wearable devices would have lower barriers for clinical settings. Anything that's invasive or minimally invasive will have slightly higher, not slightly, will have higher barriers for adoption in clinical settings.
Okay, and these are which class of devices? Like we were discussing yesterday. Just to recap,
that one would be again still be considered as a class 2 device.
Class
2. It depends on the material that's being used, because don't forget polymers in contact with skin.
So from a regulatory perspective, it would still be class 2.
Class 2.
Right. Okay. Interesting. Okay, so we have few more minutes, so we can actually any suggestions or any points you want to add here from your side?
So talking about the ISF fluid,
yeah,
I have just one question: is the concentration and composition of ISF is all around the same in our body? Because it can vary, sir.
I mean, the skin is, as I said, uniform. The skin thickness is uniform all over the body.
Okay.
And therefore the assumption is that the subvascular plexus, the distribution of capillaries, is also uniform all over the body.
But I don't think, sir, it will be uniform composition.
You're talking about the superficial. You're talking about the extremities of the body. See, you shouldn't be confused between distribution of blood vessels in the organ and in the skin. Skin is definitely an organ, but the distribution of the subvascular plexus is much more. So if you're looking at, if your assumption is based on the color of skin being different in different parts of the body, that's related to your melanocytes. So the melanocytes area which are exposed to the that are more exposed, that's part of the skin is more darker in color. That's not got to do anything with the blood distribution. The subvascular. So what I'm trying to say here is the ISF is in constant equilibration with the subvascular plexus, the very fine capillaries, right? And this distribution of subvascular plexus is, at the moment, the hypothesis is that it's constant all over the entire body. So if I did a measurement on my forearm or my back arm, it shouldn't make a big difference. It won't make a difference.
So in this PPT, do you have, just to add to his question, like so do you have an anatomy of the skin in this PPT anyway?
Unfortunately, no, this is more on epidermal, so I didn't go into the anatomy of skin. But we can discuss that when we should be having subdermal coming up again at some point. We will discuss it then.
So actually what happens is, you have blood capillaries going all over the skin close to the surface. So the analytes which are leaking out of the capillaries, blood is composed of 2 parts, right? Plasma is one of them. So whatever analytes is leaking out of the blood capillaries, it's going into the interstitial space. So this is a space which is the fluid is actually surrounding the cells, and so as long as you have capillaries in and around that space, you would have similar kind of. So that's why there is a tight correlation between blood and ISF. It's because of the capillary blood which is going there and innervating these areas.
While the sweat is produced by sweat glands.
Yeah, while as the sweat is produced by sweat glands, and
so things have to actually diffuse from the ISF into the sweat glands.
Yeah, and then from the sweat it comes out. Yeah, exactly. So, so are there any ways to artificially produce sweat, like by stimulation current?
There are different ways. I think if you did some literature review, there are different ways of getting artificial ways of producing sweat.
Okay. So I think there are some materials like some chemicals also.
Yes.
Stimulate sweat. Yeah, there are also other like papers I have also seen in which people are using.
So for all these studies, they have actually stimulated sweat. It's not natural sweat.
So
I mean, in the US, imagine the winters are very cold.
Yeah.
So you hardly get anybody to sweat on the surface. You can get sweat around the armpits, but around the surface, It's really very hard.
Yeah, and also you develop devices for all seasons. It's not for only summer that we make the device. So you have to have some artificial way to stimulate the release of sweat. So is there any link between this reverse iontophoresis and sweat analysis? So can you use reverse iontophoresis for sweat-based applications?
Reverse iontophoresis is basically getting it's, as I said, even the sweat is a diluted interstitial fluid.
Yeah.
Right. So you have your sweat glands which are producing the sweat, which is basically liquid, and it's carrying the metabolites that it's getting from the skin. And where is this coming from? So again, as I said, sweat is more a diluted version of this thing. So
It depends.
So and when you do when you do the iontophoresis side of things, you are actually pulling out the ISF, not the sweat.
Right. Yeah. Because I remember seeing some papers where they are claiming that through reverse iontophoresis they are extracting sweat.
Yeah.
But I guess it differs. We don't know exactly what it is.
Yeah, it's a concentrated version of sweat.
Yeah, absolutely. Right. So so if we were to summarize, so first you have to look at the application that you are working on. So if it is a very health related diagnostic and therapeutic application, then ISF is a better target fluid than sweat. And then if you're working for something where you want to get a rough idea of like from a wellness perspective, then you can use your sweat as well. And there are many ways to extract the ISF and the sweat. So ISF generally, we, like sir was saying, we go for subdermal devices, minimally invasive and easy to access the ISF. And there are flexible biosensors that are available, and flexible biosensors have many advantages. And the future is, as sir is saying, is to look at polymer materials which are biodegradable and good for the environment. And the barriers to clinical adoption are, of course, as you move from a non-invasive to minimally invasive device, the barriers increase, and you end up in the class 2 categorization of the regulatory side of things, and then you have to meet all these regulatory approvals. So I think that's all for this session. We will reconvene again in the next half. We'll let you know the timing shortly.
Great, thank you.